Welcome back, everyone. I am incredibly excited for today's explainer because we're doing something a little bit different. Today, we are going to master renal histology by stepping into the shoes of medical detectives. We're moving way beyond just memorizing textbook facts. We're actually going to learn how to read the microscopic landscape of the human kidney. By the end of this session, you'll be able to look at a highly complex tissue sample and instantly deduce exactly what those specific cells are doing. Okay, let's just dive right into this. I want you to consider this raw histological image as our unsolved case for the day. Now, to an untrained eye, sure, it just looks like a chaotic jumble of pink circles and purple dots surrounding a central blob. But to a pathologist, this is a highly detailed, functional map of the renal cortex. Every single shade of pink, every clear space, every tiny cellular structure, it's all a clue. Our mission today is to decode these clues so we can confidently distinguish between two major players in the kidney, the proximal convoluted tubule, or PCT, and the distal convoluted tubule, the DCT. To guide our investigation, I've put together the ultimate analysis level flashcard for us to crack. Let's look at the scenario. A pathologist views adjacent renal tubules. Tubule X has a fuzzy lumen and deeply acidophilic cytoplasm. Meanwhile, tubule Y has a wider, clear lumen and lighter cytoplasm. So, which pairing is correct? Is it A, tubule X has fewer mitochondria, B, tubule X is the PCT with high energy reabsorption and Y is the DCT? C, tubule Y is the PCT lacking microvilli, D, both lack a brush border, or E, tubule X contains the macula densa. Now, hold on. Don't worry about guessing the answer just yet. We aren't going to solve this until we've gathered all of our hard evidence. Let's look at our very first major clue from that flashcard, the term acidophilic cytoplasm. When a cell's cytoplasm takes on a deeply stained dark pink or red appearance under a microscope, we call it acidophilic. But why exactly does it look like that? Well, this deep stain is actually a direct reflection of a high organoid content. Principally, it means there's an absolute crowding of mitochondria. And you know what it means when a cell is packed full of mitochondria, right? It tells us this cell has an incredibly high energetic workload. It literally needs a massive, nonstop supply of ATP to fuel continuous cellular transport processes, like running those sodium-potassium pumps. Now, what's really fascinating here is how perfectly we can actually visualize that high energy demand. Just look at these cells of the proximal convoluted tubule. Do you notice how dark and deeply stained that cytoplasm is? That is your acidophilic staining in action, driven entirely by those abundant mitochondria. But look a little closer at the inner ring, the luminal surface facing the very center. Do you see that thick, prominent, fuzzy appearance? That is a massive brush border made of very long microvilli. This fuzzy lumen is the absolute hallmark of the PCT. So it begs the question, why does the proximal convoluted tubule need all those mitochondria and that fuzzy brush border in the first place? Because the PCT is the heavy lifter of the renal system. Its physiological reality is extreme. It's responsible for reabsorbing approximately 75% of the initial glomerular filtrate. That means the vast majority of the water, the glucose, and the amino acids your body desperately needs to reclaim are being aggressively pumped back into your bloodstream right here. Seriously, just let that number sink in for a second. 75%. To process that sheer volume of filtrate, the plasma membrane absolutely must fold into those long microvilli to maximize its surface area for optimal diffusion. And to power the continuous active transport pulling all those nutrients back, the cell is quite literally stuffed to the brim with dark staining mitochondria. Its structure is a direct, unavoidable consequence of this massive 75% workload. Okay, let's transition over to our second suspect, the distal convoluted tubule, or DCT. Because it sits much further down the nephron line, it handles a significantly lighter reabsorptive workload. And because of this, the cells are just smaller, uniform cuboidal cells. More importantly, though, they totally lack that prominent brush border. Without those long microvilli crowding up the space, the inner lumen of the DCT appears wider, much more clearly defined, and strikingly empty compared to the PCT. Plus, since it's doing way less active transport, it has fewer mitochondria, which means its cytoplasm is noticeably lighter and far less acidophilic. Let's put these two side by side to really cement this concept, because it all boils down to the fact that form follows function. On one side, we have the PCT, 75% massive reabsorption, requiring incredibly high energy, super dense organelles, and a brush border to maximize surface area. On the flip side, we have the DCT, much less tubular reabsorption, requiring fewer mitochondria, which naturally results in lighter staining and a nice, clear, unobstructed lumen. And honestly, this visual right here brilliantly illustrates exactly what we've been talking about. Just look at this direct, side-by-side comparison of the actual cells. Over on the left, the PCT cell is practically bulging. Its cytoplasm is super dark, and those tall microvilli are protruding aggressively right into the lumen. Then on the right, the DCT cell is smaller, it's lighter, and it has only short, very sparse microvilli. As a pathologist, these visual discrepancies are your immediate signposts. You literally don't need to guess. The cells are showing you their job descriptions. So, the crucial point is taking all of this evidence and applying it back to our unsolved case. Let's return to our flashcard and systematically eliminate the wrong options based on what we now know for sure. Option A claims tubule X, the fuzzy one, has fewer mitochondria. We know that's completely backwards. The fuzzy PCT is packed with them, eliminated. Option C claims Y, the clear one, is the PCT. Wrong again. The PCT is always fuzzy, eliminated. Option D says both lack brush borders. Obviously false. What about E? It claims tubule X, the PCT, contains the macula densa. But our source material tells us the macula densa is actually a specialized region of the DCT, so that's eliminated. That leaves us with option B. Tubule X with the fuzzy, dark cytoplasm is the high-energy PCT, and tubule Y with the clear lumen is the DCT. This is spot on. Let's do a super quick recap of the analytical deduction steps we used to arrive there, because this is exactly how you want to approach any histology image going forward. Step 1, you look for the lumen. If it's fuzzy, you've instantly identified the PCT's brush border. Step 2, you look at the color intensity. If it's deeply acidophilic, you immediately link that to the PCT's high mitochondria count. Step 3, you look for contrast. If the neighboring tubule has a wider, clearer lumen with a noticeably lighter stain, you've confidently confirmed it is the DCT. It's just a simple, reliable, 3-step visual checklist. Ultimately, everything we've uncovered today stems directly from this core thesis from our source material. The reabsorptive function is reflected in the structure of the epithelial lining. Histology isn't just a random collection of pretty stained patterns on a slide. It is a direct, microscopic snapshot of a living cell hard at work. The workload literally shapes the physical architecture of the tissue itself. Which leads me to leave you with this final, somewhat provocative thought. We've seen how the kidney's tubules physically alter their cellular appearance, packing in extra mitochondria and growing huge microvilli, based purely on their metabolic workload. So, if this fundamental rule of biology holds true, how might cells in other high-demand organs, like the constantly beating heart or the chemical processing liver, adapt their own microscopic form to survive their unique workloads? Definitely keep that in mind the next time you look at a tissue slide. Thank you so much for joining me for this explainer, and as always, keep learning.